And gradually, we've been kind of knocking at the door of higher and higher tech stuff to manipulate the actual DNA that causes those traits, rather than just breed things together to get those traits. Why not just go into the cells and mess with them? And that's definitely where we are now, in ways that surprise even me, having taught about this for as many years as I have. The first thing you probably need to know about to understand genetic engineering technology, it's easy enough to understand getting DNA out of a cell. You really just have to shake the cells up with some soap. Soap is very good at disrupting cell membranes because of what they're made of. Detergent like busts cell membranes and nuclear membranes right up. Then you need a way of separating the DNA from everything else, which is pretty easy. We are probably going to do it with strawberries in a couple of days. All right, just isolate the DNA. You've got the DNA. Great. But the DNA, there's 6 feet of your DNA in every single nucleus. That's not a small enough bit to work with, right? We need a way to isolate actual genes. And that's where restriction enzymes come in. Restriction enzymes are basically your DNA scissors. Huh?
That smells so good.
Yeah. So by DNA scissors, of course, I don't mean they're actual scissors. They're enzymes. Remember that enzymes are special proteins that cause particular chemical reactions to happen. You can just buy restriction enzymes from a biological supply company. New England Biological Supply, I think, is the biggest one. So I'll look them up and show you. New England Biolabs, yeah. Restriction endonucleases, that's a fancier term. There's literally hundreds of different ones. Oh, so you can do this at home. And it's even easier than this, but this is like, 15-year-old biotech. Come on, let me. Okay, I'll get it pulled up when it wants to come up. But basically, you order these things, you spend a couple $100, and you get a big package that's full of dry ice and a little vial like this with maybe a half a centimeter of liquid in the bottom. And that's your restriction enzyme. And what you're going to do is take the full DNA sample, probably a bunch of copies of the full 6 feet of DNA from a human cell, and you're going to mix it up with this, and it's going to cut that DNA up. That's the basic idea. So there's hundreds of these enzymes. They all cut DNA slightly differently. They all look for a particular sequence of A's, T's, G's, and C's.
Have you ever bought these?
Yeah. Product listing. Here they are. So this is just the A's. They all have weird names based on what their creators wanted to name them. A's, B's. We're going to focus on of them. Eco R1 is the second one, and Heinz 2 or Heinz 3 is the first one. Just as examples, and these are 2 of the first that were ever made. If you're interested in kind of what it's like, click on one of these. To click on one, I just want prices. I just want you to see what the price is. Anyway, oh yeah, definitely. Probably just based on how hard they are to make. It's killing me. Really should just be tapping on the screen, shouldn't I? Anyway, a couple $100. 1,500 units is $287. It's 20,000 units per milliliter, so 1,500 units is like an eighth of a milliliter. So a tiny amount of liquid that you're getting. It's a little bit scary when that kind of thing comes in the mail and you realize what you've spent your money on. Anyway, we're going to look at two examples. Example one being Hind 2, which is the simple one. Which basically locks onto your DNA and spins along the double helix and stops anywhere it finds GTCGAC. And when it finds that, it cuts. And when it cuts in the simplest way possible. It cuts straight down the middle between the C and the G and the G and the C. So you get two pieces of DNA that have been cut right there. It's called a straight cut. Very effective for lots of things, including DNA fingerprinting, which we'll talk about soon. Lots of uses for it. What it's not good for is the kind of sci-fi stuff that we started talking about yesterday, the kind of stuff like taking a gene from one critter and sticking it into the DNA of another critter, right? Transgenic type things where we're transferring genes from one critter to another. That requires a slightly more fancy restriction enzyme like EcoR1. EcoR1 similarly just looks for a particular sequence of DNA. By the way, no, you don't have to memorize these sequences. I'll always tell you what the sequence is. But when it finds that sequence, it cuts in a slightly different way. It cuts crooked. It cuts a kind of zigzag pattern that leaves what we call sticky ends. Why does that matter? Well, it matters because those ends, and keep in mind this is a much longer DNA strand we're talking about, and we've just cut it in one spot, and it's left the end of each big piece with a dangling TTAA and a dangling AATT. If you leave those floating around together, there's a possibility they might stick right back together. Because remember, those complementary bases form bonds with each other. But here's the thing. Any two pieces of DNA, doesn't matter what critter they're from, any two pieces of DNA that have those sticky ends can stick back together. So those sticky ends are the basis of the original recombinant DNA technology. In other words, getting DNA to recombine with other DNA to put genes that come from one critter into another critter. This is the first step of what you're going to know how to do by the end of this chapter, which is to make a glow-in-the-dark cat.
Are those real?
Yeah. Okay. Glow-in-the-dark cat.
Who just thought of that? They're just like, I wish I could see you at the dark. You just like paint it and glow stick into the inside of the
Yeah, it was
Yeah, that's fake. This is real. That's the real glow-in-the-dark cat. All you got to do is splice the DNA that makes another critter glow, for example, a firefly. or bioluminescent sea life, and splice that gene along with a couple other genes into the cat. Well, you got to clone the cat too. That's the thing. The trick is you got to put the gene, if you want the gene in every cell in the cat's body, you got to get to that first cell before it becomes the cat. And the way to do that is clone, right? So that's where we're going. You're going to know how to clone a cat and make it glow in the dark by the end of this chapter. Just to give you a sense.
Videos of it glowing.
We can probably find one. Anyway, let's look at EcoR1 first. Nope, that's not. Thank you. There. Here's what EcoR looks like at work on the molecular level. Hey, yo.
Tool used to cut DNA is a friction enzyme such as E1. The enzyme has a precise shape that allows it to run along the groove of the double helix. Adding, in the case of EcoR1, to the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted.
Oh, that's
When
way
it cuts,
mistimed. That's what that is.
uses
Interesting. Let's mute that because it's way off. And I'll be your narrator. This is the bacterial loop of DNA, just for simplicity's sake. That's the restriction enzyme. That's EcoR1. It spins along the DNA until it finds its particular bases it's looking for, GAATTC, and it cuts. But it cuts in a way that anything else that was cut with the same enzyme has sticky ends that'll recombine with it. So that's how you get a gene into something else from something else. Okay? Now, that is one thing. But just cutting it's not enough. You also got to be able to sort it. For which you need something called gel electrophoresis. And stay with us. Gel electrophoresis is actually, I mean, it's, you got to make Jello. You make some Jello, and you use this equipment I got up here. And I can't afford restriction enzymes for you guys to work with, or the real Jello they work with. So I bought some weird stuff from Thailand. It's called Telephone brand agar powder. Oh. I don't know. It makes some kind of edible Jello kind of thing with it, but it totally works for our purposes. So at some point I'll get this cranked up for you. But yeah, you basically mix it up like you're making Jello. You boil some water and you dissolve the powder in it, and then you pour it into one of these, which is just like a little tub. It's maybe about a centimeter deep. It's got these bumpers that are making it like sort of like a pool for your Jello. And you pour it, to that centimeter level, and then you put this comb in it, which makes little indentations in it. Let me show you. This is what your homework's going to be about. So you pour that gel and you put the comb like the comb makes these little holes at the top of the gel. All right. And then after you've cut up the DNA with restriction enzymes, you inject the samples of cut up pieces of DNA into the wells at the top of the gel with one of those little micro pipettes. And you put it in this big tub full of a buffer solution that conducts electricity. And you close up the tub and then you plug it into the power supply. And the thing is, DNA is slightly negative in charge because of its phosphate group that's negative. And so if you set the charge up right with the positive end at this end and the negative end at this end, it drags the DNA through the jello. This is so weird, I know. It drags the DNA through the jello. And the little pieces of DNA move faster through the jello than the big pieces, because the big pieces get more resistance from being dragged through that stuff, right? And so the sample separates out by size. You're basically sorting the DNA pieces by size. You put some dye in there that you loaded initially so that you know when it's getting toward the end, but the DNA is invisible at this point, and then you have to stain it. So you pull the strip of gel out and you dunk it in this stain, and you can see where the samples have moved to. That's the basics, okay? I'll just write some of that down. So you pour the gel, you inject the fragments at the top of the gel, you run a current through the gel.
Yeah. Wow.
To make a glow-in-the-dark cat.
Dude, what is, just like,
No. So that's a, we did it because we could example. The example that resulted in a multi-billion-dollar corporation is they took the gene for human insulin, which of course is this product that diabetics need, type 1 diabetics, right? That we used to have to crush up cow pancreases for. They took EcoR1 and snipped the human gene for insulin out of a human and put it in a bacterium. And now all you need is a big vat with bacterial nutrients, and you seed those bacteria with the insulin gene in them, and that vat puts out gallons of insulin every day. Genentech is the name of the company that basically invented bacterially produced insulin. So that's why. That's the simplest example of why. It gets more complicated from there to where I'm going to show you a video of transgenic goats that produce medicine, but only in their milk. And you can just drink the milk and get the medicine. Armani. So they only needed one cell from the guy they took it from, is the thing, right? Because you've got the same genes in every cell in your body. That's the easy thing to forget. That's a good question, is that like these genes you have, they're copied in every cell, every nucleus of every cell. So, okay, you're sorting these pieces that you cut by running this electric current through the gel. The DNA fragments get dragged toward the bottom of the gel, and the smaller fragments move faster than the bigger ones. So you're sorting them out, and then you have to stain it because the DNA is basically clear and very small. So once you stain it, you can see where the bands are. Maybe that's all you wanted, was just to see what sizes those pieces were. And you can create a scale that shows you, okay, it moved this far, so that means it's this many base pairs long. Or maybe you're after one of those pieces, and if you can identify which piece you want, you can suck it right out of that gel and use it.
you need like a license to do this or something, or you can just like buy stuff and then make glow in the dark cat?
So yeah, that question of like, what are you allowed to do is the big question. And you know, groups of scientists and lawmakers have been trying to figure that out for years now. to where we know that you can do more than you should be allowed to do, but where do you draw the line? And we'll definitely be talking about that. Because the possibilities go into like movies you've seen, like, did you ever see The Island? Oh, no. There's one that you have that you do know. Red, something about the red scorpion.
Red.
No, like it's about rich people growing their own clones as organ donors. House of the Scorpion. Okay, yeah. So that's certainly possible, but obviously not something that should be allowed, right? So most people can agree on that line, but as you get closer to like, should you be able to edit babies' genes if they have genetic disorders that could kill them? Ooh, that one tests a lot of people. So we're gonna draw our own personal lines here in the very near future. But these are the most basic tools of modern genetic engineering. Cut the DNA up, you sort the DNA with the gel, and then if you want to mess with the pieces, there's some more steps you got to take. But the basic procedure is cut and then sort. Restriction enzymes and then electrophoresis. Questions? Okay, you're going to need a device for this activity. It's basically a little genetic engineering web quest, or gel electrophoresis at least. Right, with the website that's included there, this should not take a super long time. It is due tomorrow. Yes, sir. That's because I don't like you. I can post the link if you don't feel like typing it out. Give me a second.